Thank you, Professor Park. So it is great to meet you here. And many thanks for inviting me this GMI SNU International Symposium. And today I'd like to talk about the mitochondrial DNA mosaicism in human somatic cells, which is mostly unpublished at the moment. Just a moment. Okay. So can you see my pointer?
Yes, I can see that.
Okay. So my laboratory is looking at somatic mosaicism. It is, simply speaking, it is we are revealing many genomes in one body. So more simply, we are looking at the genomic difference between two cells in an individual. So we have around, in one body, there are around 40 trillion of cells, and their genomes are slightly different due to the post-zygotic or somatic mutations after birth. So there are many questions on somatic mosaicism. For example, what is the basic rate of somatic mutation in your body? Or there must be some individual differences, cell type specificity, and also the mutation rate can be uniform, but sometimes it can be accumulated a lot of mutation in one time. So we are wondering the rate of somatic mutations in our body. And also we are also interested in some mutation types as well. So most of the studies are talking about point mutations or base substitutions or indels, but there must be some copy number changes, genomic rearrangements, or rapid mobile elements jumping, for example. So we are wondering the mutation rate on the non-point mutation variants in the genome. Also, lastly, the functional impact of somatic mosaicism on health would be another important question. So for the last 5 to 10 years, many research groups have analyzed the somatic mosaicism in the body. And if you can read the review paper by Inigo Martin Corena, you can understand that this field very well. So this is very interesting, the figures. So for example, these figures show the skin of 3 different individuals. And as you can see here, in the skin of old individual has many clones, which are amplified from single cells. And the clones have acquired many mutations. Somatically acquired many mutations. So we can say in the old tissue, there are many somatic mutations and many more winner clones that are expanded from single cells. So we are trying to analyze the somatic mutation in single cells, and in theory, the study is very straightforward, very easy to conduct. So we can collect many single cells from a body, and we can conduct genome sequencing of those single cells, and we can conduct bioreference analysis. And finally, we can identify the somatic mutations acquired in the single cells. But in reality, it is not that simple because, as you can see here, each of the single cell acquires their own somatic mutations mostly. And because the signal-to ratio of each mutation is very low, so if we conduct bulk tissue sequencing, we hardly find any mutations from the bulk tissues. We can conduct some single genome amplification sequencing using PCR or MDA or PTA, but their signal-to-noise ratio is still very low, and we find so many false positives from the whole genome amplified whole genome sequencing. We can conduct our other targeting strategies using, for example, duplex DNA sequencing, but it's good to understand the overall mutation rate, but the size of each clone cannot be determined by these targeting strategies. So what we do in our lab is we collect many living single cells from a body, and we expand those cells into some colonies, and we do whole genome sequencing without whole genome amplification of the DNA. So using this method, we can detect this method has pros and cons. So the advantage is we can detect somatic mutations very clearly. But the clonalization step is very labor intensive, and the speed is quite low, and it is not as scalable. So, but anyway, we wanted to detect somatic mutation very clearly. So we are using the clonal expansion and whole genome sequencing method. Now, previously we analyzed nuclear genomes to detect somatic mutations or somatic mosaicism in the nuclear genome. But recently we are looking at the somatic mutations acquired in mitochondrial DNA. As you may already know, the human cells, somatic cells, have the mitochondrial DNA. And the mitochondrial DNA is a circular 16 kb long DNA. There are around 13 protein coding genes, and also 2 RNAs and 22 tRNAs. So because this genome is much shorter than nuclear genome, so it can be easier to analyze the mutation, somatically acquired mutation in mitochondria. But sometimes it is more challenging to understand or to detect the mitochondrial somatic mutation, because in each cell there is around 1,000 copies of mitochondrial DNA. So many mitochondrial mutations are below the detection threshold by whole genome sequencing. For example, if there is only one mitochondria has a mutation out of 1,000 copies, the variant fraction of the mutation is only 0.1%. So it is below the detection threshold. So this mutation could not be identified by genome sequencing. We can identify mitochondrial DNA mutation only when the mitochondrial DNA are somehow expanded from the single cell mitochondrial DNA pool. So it is the challenging part of the DNA, the mutation analysis in mitochondrial DNA. So from cancer, many people were interested in the mitochondrial DNA mutation in cancers because in some Warburg effect, and many thinks that mitochondria are inactive in human cancers. So many people conducted the mitochondrial DNA mutation analysis. So around 10 years ago and 3 years ago, we conducted the mitochondrial genome analysis from the cancers. But this study is to analyze the mitochondrial mutations in human normal cells. So to this end, just a moment there. Just a moment. Okay, the chatting is not for me. Okay, so to detect a mitochondrial DNA mutation in single cell, firstly, we clonally expanded some single cells from 3 different tissues. So from the colon crypt, colorectal epithelium, and fibroblast, and hematopoietic stem cells. So from the expanded clones, we conducted whole genome sequencing of the clones, and also whenever available, we combined RNA sequencing from the single cells, and we analyzed the mitochondrial DNA mutation from each of the clone, and we compared the mutation with the RNA sequencing data. So the coverage of the mitochondrial DNA sequencing is over 7,000x, so it is quite high. So we can detect many mitochondrial mutations quite sensitively. And the mitochondrial detection variant fraction threshold was over around 0.3%. But our cutoff was some local specific. So for example, in the left side, as you can see here, this is the mitochondrial site 1,197. And as you can see here, the sequencing error rate from the region is very, very low. So we can reduce the cutoff the threshold to 0.005. Is the very low variant refraction threshold. But sometimes in the error-prone the mitochondrial side, we had to increase the detection threshold. So the detection threshold can be different locus by locus, but overall it was 0.3%. So from the around 2,000 normal clones, we found around 6,400 mitochondrial DNA mutations. So at the face value, in each clone, we found around 3 mitochondrial mutations. But as you can see here, there are many interclonal variability. So for example, in this fibroblast clone, we detected around 15 mitochondrial DNA mutations from the mitochondrial DNA sequences. But some other clone, we detected only 0 or 1 mitochondrial DNA mutations. Also, the variantal refraction of mitochondrial DNA mutations were also highly variable, from mostly the homoplasmic variant to the variants showing very low variantal refraction. What about the mutational spectrum? So it is very interesting that the mitochondrial DNA mutations are, the vast majority of mitochondrial mutations are C2T or T2C mutation. But the more interesting thing is the C2T, the wild type base of C, of the C2T mutations are located mostly in the heavy strand of mitochondrial DNA. And the T2C, the wild type T bases are located in the light strand of mitochondrial DNA. In other words, we can find the high asymmetry, high level of the asymmetry of the mitochondrial DNA mutations. So which is coupled with the mitochondrial DNA strand. So the mitochondrial DNA mutation spectrum is quite similar, slightly different, but the C2T and T2C dominance is widely used from 3 different tissues. So more or less, their mutational processes would be quite similar. As you can see here, in the D loop region, or in the vicinity of the mitochondrial origin of DNA replication, the mutation spectrum the asymmetry is quite reversed. So this finding shows again that mitochondrial DNA mutations are tightly coupled with mitochondrial DNA replication process, because in the D loop region, the mitochondrial DNA replication process is somewhat bidirectional. So from this mutation spectrum, we can understand that the mitochondrial DNA mutation process is very tightly coupled with mitochondrial DNA replication process. And sometimes we find the highly localized hypermutations of mitochondrial DNA mutation. It is similar to cuttages in the nuclear genome, the mutation. So we can understand that sometimes the mitochondrial mutations some mitochondrial DNA are hypermutated in our somatic cells. Okay, so now we analyze the mitochondrial DNA a little bit deeper. So, for example, in the individual of DB2, we sequence 27 clones from her skin. So around 50% of the clone, we found the same mitochondrial DNA mutations. So this is 16,400 C2T mutation. So around 50% of the clones, those clones carried that mutation. But interestingly, the mutations are mostly absent in the other 2,000 clones derived from around 30 individuals. So this mutation is highly enriched in this specific individual. How is that possible? So one scenario is that mutations, this same mutation was acquired in this individual again and again, repetitively. But that is, we think that is unlikely. So other more plausible scenario is the mutation was present in the first cell of the individual, or the mutation was some heteroplasmic mutation in the fertilized egg, and that is why the mutations are seen in many different clones of the individual. Similarly, we found the somehow recurrent mutations only found in the specific individual. It is another example of some mutations acquired in the fertilized egg. So we think that kind of mutations, the highly enriched in one individual and not found in the other individuals, are mutations that are present in the fertilized egg of the individual. And we named the mutation as HET-FV mutations. So fertilized egg, if the fertilized egg have the mutations in the first cell of our life, then after the cell differentiation, they will appear in many different clones repetitively. So we think using our analysis, we can detect many mutations that are acquired in the fertilized egg. And one interesting characteristics of the mutation those fertilized egg mitochondrial mutations should appear in many different cell types. So for the mutations in fertilized egg, we questioned whether they are found in many different cell types. So as you can see here, the same mutations were found in the fibroblast clones, as well as in the blood polyclonal blood cells as well, and their variant refraction is more or less similar. From the example, in the clones, the variant refraction, the clone average variant refraction is 14%, and in the polyclonal blood, it was shown around 15%. So if we compare the clone average, the variant refraction, and the variant refraction in the blood, they show very strong linear correlation. So we believe that the clone average variant refraction, or KVAV, is the value shows the initial heteroplasmy level of that mitochondrial mutation in the fertilized. So from the 31 individuals, we can identify the landscape of the mitochondrial heteroplasmy in the fertilized egg, and we found that most of the fertilized egg have the mitochondrial heteroplasmic mutation initially. So this is the landscape. So as you can see here, sometimes in the fertilized egg, there are the highly heteroplasmic DNA mutations. For example, in this individual, show the heteroplasmy in the fertilized egg over 30%, sometimes over 16%, 8%, and so on. So our conventional wisdom is that the mitochondrial DNA in the fertilized egg is very pure, but in reality, there are many heteroplasmy even in the fertilized egg in our cell. Another interesting thing in the fertilized egg variant is their variant refraction in the clones are highly purified. Let's say the clone average, the variant refraction is around 20%. then we can expect that in each cell, we can expect a 20% of variant refraction of the mitochondrial mutation. But in reality, as you can see here in the left side, the mutations are highly purified, meaning that we find mostly homoplasmy or simply wild type. So how can this happen? So we find many mitochondrial mutations in the heteroplasmy, and again, collectively they show this U shape. So almost a homoplasmy or almost 0% of the variant refraction in the somatic cells. So the possible mechanism, there are two possible mechanisms for this. One is the early bottleneck. So in the cleavage of the human embryogenesis, mitochondrial DNA replication doesn't start at some specific stage. And so mitochondrial DNA copy numbers per cell is highly reduced. So if the bottleneck size is around 1 or 2, then the only 1 or 2 mitochondria can be somehow selected or drifted in the bottleneck. So purification can happen. Also, another possible scenario is the lifetime drift. So for the entire life, so the mitochondrial DNA should be somehow drifted. So for example, in every cell division, there should be some unequal mitochondrial DNA segregation. Also, some mitochondrial DNA should be damaged, and their damaged mitochondria should be repaired somehow. So during that kind of process, there should be a lot of mitochondrial lifetime drift. So these are two different non-exclusive scenario, and we wondered which scenario is more correct. So as you can see here, the purification is stronger in old somatic cells. So in the bottom, as you can see in the blue, we collected many clones from 19-week, the fetus of the abortus of the embryo. And for the fertilized egg mutations, as you can see here, we do not find that kind of the purification of mitochondrial DNA mutation in the clone. But in the old individual, the clones from old individuals, we always find that kind of purification. So from another systematic analysis, the fixation index shows a strong linear correlation with the age. So it suggests that the early mitochondrial bottleneck is not the main mechanism for the purification, but the lifetime drift is the main mechanism. So there are two different processes underlying the mitochondrial DNA drift. And we think that the mitochondrial, this kind of lifetime turnover is the main mechanism of the purification. So from the landscape of fertilized egg mutations, we can calculate the mitochondrial turnover rate in our 3 different tissues. So in colon, the mitochondrial turnover rate is 22 per year, and the fibroblast is 42 turnover per year. So it is what we could calculate from our analysis. So the first part of my talk was about the fertilized egg mitochondrial mutation. So firstly, we revealed that mitochondrial mutations are quite, mitochondrial heteroplasmy is quite common in the fertilized egg, and using the fertilized egg mitochondrial mutations, we could calculate the mitochondrial turnover rate in different tissues. The second part is the sporadic mutations. So in sporadic mutation, the mutations were not present in the fertilized egg, but in some stage of lifetime, it should be acquired in only one mitochondrial DNA, and it should be expanded to be detected in our the sequencing. So there are two main components are operative in the sporadic mutations. One is the basing mutation rate and mitochondrial turnover rate. So usually, because these two factors are combined together, so it is very hard to decompose those two components. But because we already calculated the mitochondrial DNA turnover rate from the fertilized egg mitochondrial mutation, now we can detect the absolute mutation rate of mitochondrial DNA. So sporadic mutation is quite difficult to detect from the clone, because as I mentioned earlier, it should be amplified to be detected in our the single cell, the genome sequencing. So as you can see here, this in the picture, there are 1, 2, 3, 4 different mitochondrial mutations. But usually, if the mitochondrial DNAs are expanded, then those mutations are undetectable. So how can we measure the mitochondrial number, mitochondrial DNA mutation number? We can say, in this picture, we can say the mutation number is 4, but it is practically, it is not very good because many of DNAs are undetectable. Or we can count the variant fraction of a top mutant. In this case, is 20%. Or we can use variant fraction of all the detectable mutations as well. So as you can see here, we see that the sum of all variant fraction of detectable mutations is a good measurement for the mitochondrial mutation in the somatic cells. And as you can see here, the older cells shows higher the sum of all variant allele fractions, or SVAF. So it shows the very strong linear correlation with the age. So we believe the sum of VAF, so SVAF, is another molecular clock in our somatic cells. So as I mentioned earlier, because we already identified the mitochondrial DNA turnover number, now we can calculate the absolute mitochondrial DNA mutation rate using our mitochondrial DNA landscape. And the mutation rate is 10 to the minus 7, interestingly, which is very similar to the mutation rate of polymerase gamma, which is responsible for mitochondrial DNA replication. So if the mitochondrial DNA copy number is around 1,000 in each somatic cell, then in one mitochondrial turnover, we can say 0.8 new mutations can be acquired in each mitochondrial DNA turnover. So this using those two values, now we can illustrate, or we can explain very well about the mitochondrial mutations in our somatic cells. So those two components are mutation rate and also turnover rate. So mutation rate is around 10 to the minus 7, and the turnover rate is tissue specific, but it is between 22 and 42. There are some hotspot mutations. We identified there are 32 mitochondrial loci that show the recurrent sporadic mutations. And as you can see here, they are found in multiple individuals and in multiple clones. So some of the loci show the age relationship, correlation with the age, or sometimes some mutations are frequently found in the clones which were exposed to the UV light. So that means UV light can make some mitochondrial DNA mutation in some mitochondrial DNA hotspot repeatedly. So those mutation hotspot regions are in the D loop region or in the homopolymeric region of the mitochondrial DNA. Lastly, the functional consequences. So we find in the DNDS ratio analysis, which is the numerical ratio analysis between synonymous and non-synonymous mutation, and we don't find there are the strong selection, positive or negative selection in missense mutation or truncating mutation. That means the acquisition of mitochondrial mutations are mostly neutral. But in terms of mitochondrial DNA variant fraction in the cell, we can find that truncating mutations show much lower the variant fraction in the cell. That means clonal expansion of mitochondria carrying truncating mutations are not negatively selected in the cellular lifetime. So it means acquisition is mostly neutral, but the clonal expansion of mitochondria can be negatively selected in our somatic cells. DNA and RNA comparison, most of the DNA and RNA show similar variant fraction of mitochondrial DNA, but some tRNA mutations and RNA mutations show some discordant variant fraction of the variant fraction. So it means that tRNA mutation and RNA mutation, sometimes they are associated with the stability of mitochondrial RNA in our somatic cells. In cancer, we find more mutations and the more SBAF values. That means a higher number of truncating mutations. So that means the mitochondrial mutation turnover rate can be increased during tumorigenesis during the tumorigenesis of the somatic cells. Mitochondrial copy number, we find very huge interclonal the variability of mitochondrial copy number. So on average, it is between 500 to 1,000. But as you can see in the fibroblasts, for example, some clones show 200 mitochondrial DNA copy. Some clones show more than 2,000 mitochondrial DNA copies. So it shows a huge variation. But in terms of RNA gene expression level of mitochondrial DNA gene, they are almost similar across the clones. So that means mitochondrial DNA copy number is not a major component that control the mitochondrial RNA expression. Sometimes mitochondrial DNA structural variations are found. We found 2 structural variations out of 2,000 clones. So the rate of mitochondrial structural variation is around 0.1%. So the summary is quite straightforward. The mitochondrial DNA heteroplasmy is quite widespread in human fertilized egg, and we could calculate the mitochondrial DNA turnover rate using the fertilized egg mitochondrial DNA mutation landscape. Also, we could calculate the absolute mitochondrial DNA mutation rate using our landscape. So this is what we observed from the mitochondrial DNA sequence analysis. Sequences from 2,000 clones, and the manuscript is the preprint was posted a week ago in the research scale, and the manuscript is under review at the moment. So the most of genome analysis were conducted by Ji-sung An, a talented PhD student in my lab. And there are many, I'd like to thank all my lab members and also collaborators who contributed the samples and did their best to clonalize those single cells in our lab. And that's it. Many thanks for your attention, and I'm happy to take any question. Thank you.
Thank you, Professor Ju, about the mitochondrial mutations. And if you have any questions, please type in the Q&A form. And actually, there's one question coming in. So the question is, thank you for your valuable talk. I have a question about the accuracy of VAF. What is the most critical factor that leads to the differences in VAF according to the locus?
So it is the sequencing, the sequences uniqueness of the sequences. So in the homopolymer regions or in the repetitive sequences, usually the background mutation rate is quite high. So in that case, we had to increase our cutoff of variant refraction quite high. But if the sequence was quite unique and the sequence diversity is quite high, in that case, in some loci, we found that the background error rate is extremely low. But in sometimes, even though the sequence is quite unique and there is a huge diversity, even that case, we found in some cases the background mutation rate is quite high. So we have the error metrics for all the mitochondrial mutation, mitochondria loci, but it would be very interesting. We can analyze the underlying features that control the background mutation rate.
Okay, and the next question is how stable the clones are during the clonal expansion process?
Yes, that is another good question. So we can how can I say? We can check the stability of genome sequencing in mitochondrial DNA mutation detection through serial culture of the clones. So we conducted that kind of analysis for a few clones, and around 93% of the mitochondrial mutations were highly correlating in longitudinal clones. Around 7% were absent or newly appeared. So, but we believe those are not the sequencing or culture artifact, but we believe that some mitochondrial mutations near the VAF cutoff can be identified or undetected in the different genome sequences during the serial culture. So at least 90%. So simple answer is 93% of the mutations are recurrent in our serial culture.
And the last question is, how does the mitochondrial mutation influence the cell phenotype, and vice versa, how the cell phenotype can influence the mitochondrial DNA mutation profiles? And another question is, how this defect actually inherit from mother to child?
So the first question is a quite important question, but unfortunately, we do not find strong correlation between mitochondrial DNA mutations and its cellular phenotype. So in some clones we found, some clones are carrying the truncating mutations. Sometimes their variant fraction is quite high, 40% or 50%. And when we compare their gene expression profile against the cells without the mutation, surprisingly, we don't find that many differentially expressed genes. So we believe that the truncating mutations below some certain threshold, it does not affect the cellular phenotype. Mostly they're neutral. So that is what we believe, what we observe. But as I briefly mentioned, the tRNA mutations, some tRNA mutations or RNA mutations, they are changing the stability of the transcript, mitochondrial RNA transcript. So this is quite an interesting part to be further analyzed in the next study. And inherited to mother to child, probably it is about the germline. So we analyzed the new mitochondrial mutation in the somatically acquired in the somatic lineage. But if the same mitochondrial mutation rate and mutation, the mitochondrial turnover applies to the germline, probably we can predict the emergence of new mitochondrial DNA mutations in the germline as well. But because there are a lot of bottleneck in the genesis of egg, the female germline, so it should be more difficult to answer. And probably we need many sequencing of many generations in families. That will be very helpful to understand the germline, the dynamics of mitochondrial DNA mutation in the female germline.
Okay, I think that was all the questions. So let's thank again for Professor Ju for sharing us interesting stories. And because we had really enthusiastic discussions, I think we used most of our break time. So
Sorry
without
about
any
that.
break, so yeah, we move
Apologies.
on. No apologies. So you're